Hello guys, today I will discuss amyloidosis. So firstly, what is amyloidosis? Amyloidosis is a condition that is associated with number of inherited and inflammatory disorder in which there is extracellular deposits of fibrillar proteins that are responsible for the tissue damage and functional compromise. But amyloid deposition can be intracellular. So it is not a single protein. It can be derived from the misfolding of a variety of precursor proteins. To date, sorry, this is wrong. To date, 42 proteins have been found to form amyloids and can be associated with well-defined disease. So he is Sir Rudolf Virchow. He in 1854 he looked a shiny looking area in the spleen and he thought it to be starch protein. Then he applied glucose iodine and this abnormal looking shiny area on the spleen turned brown. Then after this he applied dilute sulfuric acid to it, that it abnormal area turned blue. So he saw that this area, this protein has staining properties similar to starch, which compose of amylose and amylopectin components. So hence he named it as amyloid. But in 1859, the Frederick and Kekulé, they were the first to recognize the material found by the Virchow in the waxy spleen, and he thought it to be amyloid was most probably proteinaceous rather than the starch-like. Now, coming to the physical nature of the amyloid. Amyloid is composed of non-branching fibrils of 7 to 10 nanometer in diameter. So, all of the amyloid share a beta-pleated, highly ordered secondary structure. So, you can see the beta-pleated, highly scanned st ordered structure, and there is a groove for the attachment of the Congo red. This is Congo red, and this highly ordered cross beta plater structure is responsible for the distinct Congo red staining and birefringence of the amyloid. So we have studied the physical nature of the amyloid. Now we will study the chemical nature of the amyloid. So amyloid is 95% fibrillar proteins, 5% it has P component and other glycoproteins. So for describing the amyloid syndrome, uniformly the prefix is A, that is for amyloidosis, followed by the suffix of the abbreviation of the precursor protein. I will explain this nomenclature later. So, what is amyloid nomenclature? To describe the amyloid syndrome uniformly, as I told in the previous slide, the prefix is A, that is for amyloidosis, followed by the suffix of the abbreviation of the precursor protein. For example, AL. A is for amyloidosis, L is abbreviation of the precursor protein, that is light chain L, and the protein can be specified further like ATTRV, V for variant, A for amyloid, TTR for transthyretin, or ATTRWT, A for amyloid, TTR for transthyretin, WT for wild type form. So this is the slide which is describing the structure of the protein. Protein has primary structure, secondary structure, tertiary, and quaternary protein structure. So amyloid is cross beta pleated structure. This is amyloid. There is abnormally folded protein in the beta pleated configuration, amyloid. So when we denaturate the protein, only primary structure is preserved. That is, what is primary structure? This is the sequence of chain of amino acid. You can see this chain. This is sequence of chain of amino acids. What is secondary structure? Secondary structure can be alpha helix and beta pleated. So in secondary structure, there is hydrogen bonding of the peptide backbones. There is hydrogen bonding of the peptide bonds. In primary structure, there is only peptide bonds between the amino acids. In secondary structure, there is hydrogen bonding of the peptide backbones. What is tertiary protein structure? Tertiary protein structure is three-dimension folding pattern of the protein due to the side chain interaction. And what is a quaternary protein structure? That composed of more than one amino acid chain. You can see here there is two amino acid chains. So this is quaternary protein structure. So we have read the nomenclature of the amyloid, and we have studied the structure of the proteins. Now we have studied the physical nature of the amyloid. We have studied the chemical nature of the amyloid. Now I will discuss the pathogenesis of the amyloid. Firstly, normally the misfolded proteins, the protein which are misfolded, are degraded intracellularly in the proteasomes and extracellularly by the macrophages. What in amyloids, it appears that this quality control mechanism failed, leading to the accumulation of the misfolded protein outside the cell. So the protein that form the amyloid fall under two general categories. First are normal proteins, second one is mutant proteins. The normal proteins that have inherent tendency to fold improperly when and form the fibrils when there is increased production, and the mutant protein that are prone to misfolding and subsequent aggregations. So there are mainly three type of amyloids. First is AL protein amyloid, AA protein amyloid, and ATTR protein amyloid. So amyloid production can be due to the production of abnormal amount of normal protein, and can be due to the production of normal amount of mutant protein. These two, that is AL protein and AA protein, are due to the production of the abnormal amount of normal protein, and ATTR protein is the mutant protein that is produced in normal amount, that has capacity to aggregate. So when is AL protein amyloid is formed? When there are certain acquired mutation like IgH, immunoglobulin heavy chain translocations, etc. There is monoclonal B lymphocytes proliferation, which form the plasma cells, and there is increased production of the immunoglobulin light chain. So due to the increased production of the immunoglobulin light chain, there is limited proteolysis of these chains, and these chains aggregate to form the AL proteins. Similarly, in the chronic inflammation, there is like disorders like tuberculosis, osteomyelitis, IBDs, and some solid tumors. There is macrophage activation that stimulate the formation of interleukin 1 and 6, which further act on the liver cells and stimulate the production of SAA protein, that is acute phase reactant. Due to the increased production of SAA protein, there is a limited proteolysis of this protein, and this protein aggregate to form AA protein. So AL protein amyloid is seen in the condition like multiple myeloma, plasma cell clonal disorders, and AA protein amyloid is seen in the condition chronic inflammatory conditions. Next, the third one is ATTR protein amyloid, which produced due to the production of normal amount of mutant protein, for example, transthyretin. So there is some mutation and there is production of the mutant transthyretin which aggregate to form ATTR protein. So this is the classification of the amyloidosis. Can be systemic amyloidosis, hereditary amyloidosis, or localized. Systemic amyloidosis is further characterized into primary or immunoglobulin light chain amyloidosis, reactive or secondary, and hemodialysis associated amyloidosis. Hereditary amyloidosis is further divided into familial Mediterranean fever, familial amyloidotic neuropathy, systemic senile amyloidosis. The localized amyloidosis can be senile cerebellar, endocrine, which can be seen in the medullary CA thyroid, islets of Langerhans, and isolated atrial amyloidosis. And these are the associated diseases. These are associated proteins, and these are the precursor proteins. These are some non-immunoglobulin and non-AA amyloidosis, which can be seen in acquired sporadic or hereditary conditions. Firstly, I will discuss the systemic amyloidosis. First heading under the systemic amyloidosis is immunoglobulin light chain amyloidosis, which was earlier called as primary amyloidosis. So, these precursor protein, this is immunoglobulin light chain, and these are produced as I discussed earlier in the pathogenesis of the AL protein, which is due to the acquired mutation. There's monoclonal B cells proliferation, there is increased clonal plasma cell production, and there is increased immunoglobulin light chain, and there is AL protein production. So most of them are composed of gamma light chain. That so light chain can be kappa light chain can be present in some cases, but most of them can be gamma light chain. So they are secreted by the monoclonal population of the plasma cells, and the deposition is associated with certain plasma cell tumor like multiple myeloma or other B cell neoplasm. But not all free light chain produce amyloidogenic potential of any light chain is determined by a specific amino acid sequence. So this was amyloid light chain amyloidosis, earlier called as primary. Now this is GIT tract amyloidosis, in which the most common is AL more than AA, more than hereditary amyloidosis. So localized form of the amyloid is also known as amyloidoma. And this is the systemic amyloid organ manifestation. In various organ, amyloid deposition can occur, which can present as various symptoms like in liver, it can present as hepatomegaly, liver failure, liver rupture. GIT can be like weight loss, anorexia, melena, and kidney like proteinuria, renal failure, and in tongue macroglossia can occur. In nerves, peripheral motor neuropathy can occur. So second one is amyloid associated protein, that is AA protein. Earlier it was known as secondary amyloidosis. As I discussed earlier, it is seen in chronic inflammatory condition. So previously it was seen in chronic inflammatory condition like tuberculosis. So but now due to the treatment drugs of the tuberculosis, it is less likely seen in the Western country. Now It is more associated with rheumatoid arthritis, COPD, Crohn's disease, ulcerative colitis, and may occur association in solid tumor like renal cell carcinoma, Hodgkin lymphomas. Next one is heredofamilial familial amyloidosis. Most of them are rare and can be limited in certain geographical area. First one is familial Mediterranean fever, which is an autosomal recessive condition in which it is an autoinflammatory syndrome which associated with excessive production of cytokine interleukin 1 in response to the inflammatory stimuli. So the amyloid fibril is AA protein. As I discussed earlier, in inflammatory condition, there is macrophage activation, there is interleukin 1 and 6 production, there is stimulation of the liver for the production of SAA. So in inflammatory condition, the amyloid fibril is AA. There are attacks of fever and inflammation of the serosal surface, and the gene involved encodes a protein called pyrin. Second one in heredofamilial amyloidosis is familial amyloidotic polyneuropathy, which is an autosomal dominant condition in which there is deposition of the amyloid in the peripheral and autonomic nerves. So these fibrils are made up of mutant TTR. There are the forms type of amyloids, hereditary amyloidosis. These are the type of amyloids. For the hereditary amyloidosis, we can do the genetic testing, we can do the Sanger sequencing, NGS, and mass spectrometry. Other form of amyloidosis is localized amyloidosis. The amyloidosis can be localized in the lung, larynx, skin, urinary bladder, tongue, and the region around the eye. So this is the, you can see the gross appearance of the kidney in the amyloidosis. A large kidney with pale waxy appearance mainly you can appreciate on the waxy appearance on the cut surface, smooth waxy appearance. But laterally it can shrink due to the ischemia. But in some, not in every case, you can see this pale waxy appearance. In renal amyloidosis, it can be kidney can be grossly apparent. Grossly, it can be normal. This is the microscopy of the renal amyloidosis. We can see on the He stain in the glomerulus, you can see the pale acellular material deposition. We can see here the pale acellular material, which on Congo red staining appears as salmon pink appearance it is giving. We can see here. And the polarized microscopy, you can see the apple green birefringence. This is apple green birefringence. So there is deposition of amyloid. So there is, you can see in the interstitium, there is, this is the PS stain. In interstitium, there is PS negative material, which on the Congo red stain showing salmon pink appearance it is given. There are the different type of amyloid which can be seen in the kidney in the different condition and different compartment. Like in the end stage kidney, we see the A beta 2M. A like 2 is the third most common amyloid to affect the kidney and majorly cortical amyloid. Now these are the different type of amyloid seen in the kidney. In spleen, amyloid deposition can be in two forms, like it can give appearance, can be limited to the splenic follicles and give appearance of sago spleen, like typoica like granules on the gross inspection. It can deposit in the splenic sinus and the connective tissue framework in the right pulp. So it can give the appearance like lardaceous spleen, like fat-like spleen. We can see here there is pale acellular material deposition, pink acellular material deposition. This is red pulp, which is expanded. This is white pulp. This is periarterial lymphoid sheath. Atrophic white pulp. Expanded red pulp due to the deposition of the pale acellular material. So in sago spleen, there is involvement of the periarterial lymphoid sheath. This is the periarterial lymphoid sheath. So in the lardaceous spleen, there is deposit start in the wall of the splenic sinuses. Splenic sinuses. These are the splenic sinuses. So next is liver. The amyloid deposition occur in liver in the first in the space of Disse in between the cords of the hepatocytes, and then it encroaches the adjacent hepatic parenchyma and sinusoids. So even with the extensive involvement, the liver function is usually preserved. We can see that apple green birefringences. We can see this pink material deposition. This is the amyloid. We can see this heart. In heart, you can see this pale acellular material deposition in between the muscle fibers. This, which on the polarized microscopy we can see this apple green birefringences. So in heart, grossly it may be enlarged. There can be pale translucent and waxy, and then can be nodules seen epicardium and endocardium. Microscopy, it can be begin as a focal subendocardial accumulation. So A beta 2 is amyloid that is associated with the hemodialysis, and endocrine amyloid, like in medullary thyroid carcinoma. There are some amyloid deposition in pancreatic islet tumor. There are amyloid deposition, and there is amyloid of age that is derived from the TTR. So in brain, we see in the Alzheimer disease, there is A beta, that is intracellular amyloid, which can be detected by the IHC. This is the beta amyloid IHC, antibody in IHC. Thank you. Next, diagnosis of the amyloid we will discuss in the next video. Thank you.